All right, so the first thing we're going to talk about is a population. So populations are smaller subsets of all the individuals of a species. It's a bit of a nebulous concept what exactly defines a population. We have the human species, and there's approximately 6 billion humans that live on Earth. But the reason we don't consider this one single giant population is that there's typically restriction in the gene flow, which basically means there's restriction in mating between individuals of the population. So if you live in the United States, especially historically, you are very unlikely to ever have offspring with an individual that lives in a different country or a different continent. And really, if you live in a different city, you are very unlikely, to not have offspring with someone who lived in that city. And so kind of where the population idea comes from are groups of individuals within a similar geographic area that are essentially all mating with each other in some sort of random process. And so we also talk about a population's gene pool, and the idea of that is that all of the alleles that are present in a population. And so it's genetic information that's carried by members of a population, and most populations contain high degree of heterozygosity. So let's talk about kind of a real life example of this. So a real life example of a population are frogs in a pond. So if you have, quite a large pond, these frogs, live at different parts of the pond, but typically these frogs are able to come together and mate with each other kind of randomly. So, what we've done is we've identified every single individual frog in this pond, and so there's a total of 16 frogs that are living within this pond. So these frogs are very unlikely to mate with frogs in a different pond because there's typically a geographical difference between them. So one of the important concepts of this population is the idea that there is allele frequency and genotype frequency. And so we right now are looking at a single allele. So kind of the idea is we're looking at a single one of these in the frog, and there are 2 alternative alleles at this position. And so we have done, basically what we have talked about right here is we've gone in and we've looked at what this allele is at this position at every single chromosome in every single frog. And so because frogs are diploid, we list both of those alleles, big A, so we're just going by kind of the historical, big A, little a to designate the different alleles, but you can think about it exactly the same way as A and G. And so we go through every single frog and we actually measure what the 2 alleles are that frog is carrying for that position. And so, these frogs are genetically identical to each other at that position. They both have big A. But they are going to be, each of these frogs, even if they have the same genotype at a single position, that doesn't mean that the frogs are identical. So you can think of it something like this. So this is frog 1, and this might be a little bit redundant, but this concept is really important to understand. So I'm going to belabor the point a little bit, and this is frog 2, okay? And so if we looked at this position right here, both of these frogs would be homozygous, and so they would both, in this case, we're just calling them both big A, and big A. But these frogs, if we looked at any sort of different position, so example, if we looked at this position right here, these frogs would not be identical at this position right here. So there'd be something like this, because this is big G, and this is big A, this is AA, and so that would be, the opposite allele. So, the thing that I'm showing you right here is just one position. But one of the things we want to measure is how common each genotype is within the population, and then how many alleles are within the population. And so these are 2 really important definitions for this chapter: allele frequency and genotype frequency. And so how we measure it is, we have simply right here listed all of the frogs that are big A. So there's 5 frogs that are big A. There are 8 frogs that are heterozygate, and then there's 3 frogs that are homozygous for the opposite allele. And so we can also do this for the allele numbers. So, if we look at this frog has 2 of the big A's, 2 of the big A's, 1 of the big A, 1, 2, and we kind of go through this and we count how many big A's are within this pond. And now we simply add up this number. So 5, 6, 7, 8, 9, 10, 11, 12, 13, 15, 16, 17, 18. And so there's 18 big A's within this population. And then for the little A's, they are 14 if we did the same process right here. So if we wanted to calculate the allele frequency, we simply add these together. So there's 32 total alleles, which is, and there's 16 frogs, so this is just 16 times 2. And now we just simply divide 18 by 32, and that gives us the allele frequency of the big A, and then 14 divided by 32 is the little a. And so we define this as little p and little q. So little p equals 18 30-seconds, and then 14 30-seconds is equal to q. And so one of the things is that they have to add up to 1, and the allele frequency would be this and this, and because you have to sum to 1 in probabilities, one of the things you can always assume is p plus q is equal to 1. So genotype frequency, again, the same sort of thing. This would be 5 divided by 16, 8 divided by 16, and 3 divided by 16. So there's 3 different genotype frequencies, because you can have, the homozygous, heterozygous, and then the homozygous of the alternative allele. So these don't have names, but these also, if you add them up, have to equal to 1. So a lot of this chapter that we're going to cover is looking at allele frequencies, understanding how they change, and then understanding the relationship between the genotype frequencies and the allele frequencies. So just to have a slide that kind of goes over these important concepts. So population, you should understand what a population is, population size, and then make sure you understand what these allele frequency and genotype frequencies are. So here, what I've shown you, just to get used to the terminology, is I've shown you the allele frequencies for 4 different alleles. So I've named this A, B, C, and D. And so if we go back up here, this is A, B, C, and D. So if I calculated the allele frequency, 1 out of 5 is the alternative allele. In this case, 3 out of 5. So 3 out of 5 are red, 2 out of 5, and then 3 out of 5. So you can see I've calculated the allele frequency for 4 of these different positions, and you'll notice that all 4 of them are different. And so this is just how we show that. So we have the allele frequency for A, B, C, and D, and we've done this, for a much larger population, but we've calculated this, and so you can see that there's a huge range in possible outcomes. So in one case it's 80%, and then the other case it's less than 0.2% that actually have this allele right here. All of these sum up to 1, so P plus Q is always equal to 1, but each one of these are different. The other thing that's important to know is that you can measure these in different populations. So this could be a separate pond that has their own unique set of frogs, and so we can measure the allele frequency in this pond and also this pond. This could also be something like USA, so these are the allele frequencies if we look at all of the individuals that live in the United States, and these could be the allele frequencies of Canada. And so the important thing to take away from this slide is that the allele frequencies don't and typically are not exactly the same to each other. So this measure is specific to a given population, and you really need to be thinking about what population am I talking about. But because these allele frequencies are different in different populations, this is why you can actually infer your ancestry using this DNA information. And so this is just a technique called PCA analysis, principal component analysis, but it's a way to take a very large data set and then kind of reduce it into something that's two-dimensional that you could graph. But each one of these spots is information about the allele frequencies from individuals from different places. So the green triangle right here are from people that live in the United Kingdom. The blue square are from people that live in, I believe, Ireland. So you can see that there's a pretty large overlap in the distribution of allele frequencies in the people that are from UK and Ireland. So it can be very difficult to actually tease apart ancestry from Scotland versus Ireland because the allele frequencies are similar enough to each other that it's hard to distinguish. But if you look at from individuals that are from Norway, for example, you see that the allele frequencies are pretty different from each other. And the people from Sweden are also pretty different from each other, and Finland. So for whatever reason, these populations have been separated enough that the allele frequencies are different enough that you can actually infer information. So when you do this 23andMe, what you're doing is you're trying to see how your DNA overlaps with something like this. And so this is just specific to Europe, but you could expand this to include individuals from all the countries in the world, and the idea would be the same. So you look for you break apart your chromosome into different pieces, and then in the end you see the overlap of those chromosomes kind of within these plots sort of here, and then that's how you go into ancestry information. So again, populations have allele frequencies, and populations have different allele frequencies, and you can use that information to actually infer ancestry of kind of where you're from based upon the set of alleles that you actually carry within your own chromosomes.